Good morning, my dear students. I'm Bharti Kathuria Sehgal from the SD Vidya School, Ambala Cant. Today, we are going to start with the next chapter, Reproduction in Flowering Plants. Now, as you know, that flowers are the reproductive parts of a plant. They are the site of sexual reproduction. The flowers contain the male reproductive part, which is called androecium, and the female reproductive part, which is called gynoecium. Now, there are two types of flowers on the basis of presence of male and female reproductive part. The first ones are called as unisexual. If the flower bears only stamens, they are called staminate, or if they bear only pistil, they are called pistillate. Examples are papaya, cucumber, watermelon, and muskmelon. Another type of flowers are bisexual, which bear both male and female reproductive parts on the same flower. Examples are pea, wheat, and China rose. Now here they have shown the types of flowers which are chasmogamous and cleistogamous. Now chasmogamous and cleistogamous we'll be discussing in the next part. Now there are different types of flowers on the basis of open or being well exposed. The first one, the cleistogamous flowers. These are the ones which do not open at all. As you can see here, the cleistogamous which grow underground and do not open at all. The examples are oxalis, viola, and commelina. The next are the chasmogamous flowers. The flowers with well exposed anther and pistils are the chasmogamous flowers which are formed above the ground. Example is oxalis. Again in oxalis, so you'll be finding both chasmogamous as well as cleistogamous. So by looking at these two flowers and by knowing about them, you can clearly predict that in chasmogamous flowers, because they do not open at all, they will have only self-pollination. While in the case of chasmogamous, with well-exposed anther and pistil, they can have cross-pollination. Now we're going to talk about the other aspects, that is the different parts of the flower, as we discussed about the pistil and stamen. Pistil, as we talked, called as gynoecium, is the female reproductive part consisting of stigma, style, and ovary. While the male reproductive structure, the stamen, consists of anther and filament. There are colored whorl of leaves known as the petals and the green colored called as sepals. Now let's discuss about androecium. Androecium is the whorl of stamens. The stamen consists of the filament, which is the long and slender stalk, and the anther, which is the bilobed structure. Now filament is attached to the thalamus or to the petal. Now a typical anther is bilobed and diathecous. Now what is this diathecous? Diathecous means it contains two theca. Now when we discuss the structure, I'll be showing you what are the theca. Now these theca are separated by longitudinal groove running lengthwise. So let's discuss it here. Look at the diagram. Now once you look at the diagram, what you will come to know is you will be seeing the structure of this part that we call as the anther. Now in the anther you can see two different longitudinal these two different parts separated by the longitudinal groove. These are called as the theca. Now because there are two theca, that's why it is called as diathecous. And again, now each theca has these four lobed structure. So these are the four parts which are present in the anther, and each of them contains the pollen sacs where there is formation of pollen grains takes place. Now let's discuss about the structure of microsporangium. Now the microsporangium is surrounded by four wall layers: the epidermis, endothecium, middle layers, and the tapetum. The outer three layers, that is the epidermis, the endothelium, and the middle layers, are protective in nature. They help in dehiscence of anther to release the pollen grains, while the tapetum is the innermost layer and it provides nourishment to the developing pollen grains. The sporogenous tissue forms the center of each microsporangium. Now let's look at the structure of microsporangium. So now this is what we just discussed about, which is the structure diathecus as we call, and there are these four structures which are present, the pollen sacs, and in these pollen sacs now you can see the microsporangium. Now these microsporangium, as I told you, have four different layers: the outermost epidermis, then comes the endothelium, then the middle layers, and the innermost, the tapetum, which provides nourishment to the growing this sporogenous tissue, which contains the microspore mother cell. Now these microspore mother cells are the ones which will undergo meiosis to produce the pollen grains. Now microsporogenesis. As I just told you, the pollen mother cells undergo meiosis to help in the formation of pollen grains. Now, we'll be discussing about this. Now, microsporogenesis is the process of formation of microspore from the pollen mother cell. Each cell of the sporogenous tissue has the capacity to give rise to a tetrad. Hence, each cell is a potential pollen or PMC. Now, as you all know that after meiosis, there is production of 4 new cells. So, this is what we are calling here as tetrad. So, each cell of the sporogenous tissue can give rise to a tetrad, and each one is thus capable of producing a pollen or a PMC. As the anther matures, the microspores get detached from each other and develop into pollen grains. Now let's discuss about pollen grains. Pollen grains is the name that you must have heard a lot, but here we are going to discuss in detail about pollen grains. Now they represent the male gametes and are spherical. They have a two-layered wall. The outer layer is called as the exine, while the inner layer is called the intine. Pollen grains are actually the male gametophytes containing the male nuclei. Now, as we were discussing that they contain two layers, exine and intine. Now, exine, it's a very important question from the CBSE point of view. Exine is made up of sporopollenin. Many times question has been asked about it, that what is the special characteristic of exine, or why there is nothing which can break down sporopollenin. Now they are made up of a substances, sporopollenin, which is extremely resistant and can withstand high temperatures, acidic and alkaline conditions, and also enzymes. Now the intine, which is the inner layer in the pollen grain, they are thin and continuous and made up of cellulose and pectin. Now mature pollen grains contain two cells: the vegetative cell, which is large with irregular nucleus and contains the food reserve, and the other is called as the generative cell. So here you can see the diagram of vegetative cell and the generative cell. Now generative cell, which is small and it floats in the cytoplasm of the vegetative cell. 60% of the angiosperms, pollen grains, are shed at the two-celled stage, while in others, the generative cell undergoes mitosis to form two male gametes. This is the three-celled stage. Now, viability of pollen grains after they are shed depends upon temperature and humidity. Viability means कितने time तक उनके अंदर capability है to germinate and then produce the pollen tube. Now, this viability ranges from 30 minutes to few months. Now, pollen grains. Now, there are certain disadvantages of pollen grains. They cause allergies and they may also cause bronchial infections in some people, which leads to asthma, bronchitis, etc. Like Parthenium carrot grass is one which is a very common allergen. Now, advantages of pollen grains, as you all know that pollen grains are obviously the very important part of the reproduction, but other than this, they're also used as in the form of tablets for food supplement. And in Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Now, these are used to increase the performance of athletes and race horses. Now, this is what is a Parthenium or the carrot grass as we were talking about, and the pollen grains of this carrot grass are highly allergic. Now, cryopreservation, you must have heard about it earlier also, means preserving something at a very low temperature. Now, this is the process of storing seeds, tissues, gametes like sperm, ova, and other biological products for future use or research purposes at low temperature. Now, for cryopreservation, temperature of minus 196 degrees centigrade in liquid nitrogen is used for pollen grain. Now, viability, as we discussed, is time period for which the pollen grains remain live. In cereals, it is like 30 minutes. In case of wheat and rice, in families like Rosaceae and Leguminosae, it can be more. Now, here you can see the method of cryopreservation, where in a small test tube, it is being preserved in the liquid nitrogen. Pollen grains are being preserved in liquid nitrogen here. Now, gynosium and formation of female gametophyte. Now, this is the topic that we'll be covering in the next video.